This is a production of Cornell University.
I'm going to talk about what I did on my summer vacation, actually. So this is actually, I mean this in a sense that, just a few months, I'll be fully retired. And at that point, I'll be asking for the faculty to grant me emeritus status. So I have to prove that I'm still actually doing something useful. And that's what this is all about today. So I'm going to talk about a major question, which is how novelty arises. It's probably the fundamental question in evolutionary biology. And I'm going to talk about it in several ways. One is about species and how they arise and evolve. Secondly, how structures evolve and how they originate. And thirdly, at the cell level, how do cell types evolve? So all three of those. So I'll tell three short stories, essentially. The first is about speciation and its consequences. Turn that one off, okay. And in particular, a couple of short stories about soybean relatives, which I've worked on for years, and also rice. And then after that, the evolution of nodulation symbioses, and finally talking about gene duplications and the evolution of cell type diversity. What these things have in common, other than novelty, is polyploidy. And Susheng mentioned that my research has been about polyploidy, whole genome duplication, for many, many years now. So polyploidy, I think, as everybody knows here, is everything above the level of two sets of chromosomes in a cell. It's a phenomenon that is very common in nature at the organismal level. Endopolyploidy is a common phenomenon, as it mentions here in this review paper, indispensable for normal development and organ formation across various organisms, from fungi to humans and plants, of course, as well. Adrian Roeder's work on giant cells in the sepals of Arabidopsis, for example. So there's muscle cell with two nuclei, and then also it's a very common phenomenon in cancer. Cancer, as it says here, is an ugly consequence of endoreplication and polyploidization. But I'm not going to be talking about endopolyploidy today. I'll be talking about whole organism polyploidy, where whole organisms are duplicated, duplicate their genomes. And polyploidy occurs throughout the entire tree of life. All of us in this room are multiply polyploid, along with all of the vertebrates, based on a very long time ago polyploidization event, all the way back here in the Cambrian explosion. Other common lab organisms, such as yeast, are polyploid. And if you like salmon, for example, and trout, you're eating tetraploids, recent tetraploids. But plants really do polyploidy in a big way. And this is from the 1,000 plant transcriptomes, 1,000 different transcriptomes of different species, among which there were 244 whole genome duplication events identified. Some shared, many of them shared, but also unique ones at the tips of these branches of the trees. And if you look over here on the side at the distribution of the number of polyploid events, whole genome duplication events in the ancestry of all of these different taxa, what you'll see is that many of the common angiosperms, so the rosids and the asterids, for example, so legumes, asters, all of those things, have an average of about 4 whole genome duplication events in their evolutionary histories that you see in the genome. So my friend Jonathan Wendell published a very nice paper on this some years back, and he calls it the wondrous cycles of polyploidy in plants. It's for us enthusiasts about polyploidy. We think of it as a wonderful sort of thing. But what we see is this cycle. Here are a whole bunch of different polyploidy events across the tree of life. And we have all sorts of events that happen on concomitants to that. And finally, this whole cycle of duplication and gene loss, biased fractionation or gene loss of different subgenomes, genome downsizing, so loss of repeated sequence, for example, rearrangements, and then you've produced a functionally diploid plant through this process. It involves many different individual processes that are called lumped together as diploidization. So this diploidization cycle, followed by another round of polyploidy, gives us the complex genomes that we see in flowering plants today. So the first of my vignettes is about speciation. I'm not going to talk much about this. This is an area where a lot of my research has been over the years. But I'm just going to mention a couple of things, excuse me, that we've been doing lately. It's a long-term project, actually, to look at the evolution of the soybean relatives that you see down here, perennial soybeans native to Australia. Jacob Landis has led much of this work in recent years, and so collaboration with Qing Li, Liu at Syngenta, and Andrew Farmer at National Center for Genome Resources in Santa Fe. This paper, I think Jacob just submitted it, what, a week and a half ago, something like that? So this was one of the main things we worked on during the summer, was understanding this legacy of polyploidy and introgression and looking at it across 2,300 different nuclear genes from genome resequencing done by Syngenta. A second project that was working on quite a bit this summer was mainly led by Zhenwei Zhu here. Zhenwei and her mentor Song Ge at the Botanical Institute in Beijing have been working on rice allopolyploids for many years. And this particular project was a genome project in which, looking at this rice that you see here, this Rhiza minuta, 48 chromosomes derived from these two diploid species here, formed around 700,000 years ago, traced the genomes using phylogenetic methods and everything. And here you'll see the circus diagram showing the subgenomes of the polyploid, and they're virtually identical in terms of synteny and their organization with the genomes of the diploid. However, when you dig a little bit deeper, you'll find that the diploidization process has started, and this paper is about how different parts of that diploidization are either episodic, some of them occurring right away after polyploid formation, and others are more gradual. For example, the acquisition of transposable elements has been a gradual process as opposed to gene loss, which has been much more episodic. So this is a project that I'm not going to say much more about, just to let you know we're working on that. So one of the questions is, why is polyploidy so common across plants particularly? And the idea is that polyploidy is a generator of novelty, both at pretty much every possible level. This classic paper by Don Levin, someone Bill knows very well, yeah, he's smiling here in the front, is a real classic in terms of all the ways that polyploidy can create evolutionary novelty. A paper by Mike Freeling a little bit later, gene balance duplications are a drive to increase morphological complexity. So this has been a recurring theme in the polyploidy literature all along. Jeremy Code, who was a student here many years ago, has been one of my top collaborators for years now. And I wrote this paper on a little essay really on autopolyploidy as an epigenetic macromutation. So a macromutation is a single event, polyploidy in this case, with many different phenotypic consequences. So it qualifies there. And epigenetics are molecules and mechanisms that can perpetuate alternative gene activity states in the context of the same DNA sequence. So this is one of many definitions of epigenetics, but it's one that suits my purposes, so I grabbed it. Basically, if you make a synthetic polyploid, just double the chromosome number using colchicine, you have a plant that's isogenic with its diploid progenitor. So it has the same DNA sequences, but it also has many phenotypic consequences when you do that. That's the macromutation, and the same DNA sequence is the epigenetic part. So an autopolyploid is an epigenetic macromutation. And that's the source of all of that novelty from the genome doubling part. I mentioned also allopolyploids, the rice, for example. And allopolyploids add another component, which is hybridity, heterosis, those kinds of effects as well. But pure polyploidy, as you might call, it has been called autopolyploidy, is a matter of just doubling the same genome, and so it by itself can create evolutionary novelty. That's the point here. Jeremy and I explored this at the cell level, trying to get caught up to cell biology, modern cell biology, and wrote this paper, and all these lines and everything show you we're talking about two different phenomena, right? Increased dosage when you double the genome, of course, and also increased bulk amount of DNA. This is something that's rather controversial, something called the nucleotype. People are familiar with genotype and phenotype. Very few people are familiar with the nucleotype, which is just correlations with various different characteristics of the plant or animal, such as cell size, nuclear volume, things like that, that follow from increasing the DNA content. And it's not clear yet at this point whether or not this is causality or correlation only, but it's an intriguing thing that you see this trend across many different traits where autopolyploids, or just plants with larger DNA contents even, show these trends of having larger cell size, nuclear volume, and those sorts of things. And this is thought in many ways to be the source of all the other phenomena that follow from that, all these different lines that I'm showing here, complex relationships. The key point of this for this talk is just that this also does lead to morphological changes, physiological changes, and even ecological changes as well. And that brings me to my next section of this talk, and that's the origin of nodulation symbioses. So nodulation is a symbiotic association between some flowering plants, a limited number of them, and diverse soil bacteria. And the bacteria are housed in these nodules, which you can see here, which then fix atmospheric nitrogen to form ammonia, so fertilizer for the plants. The nodule is a morphologically and anatomically and biochemically complex evolutionary novelty. It's not found in other plants. Its most associated nodulation is with the legume family, but it's neither confined to the legume family, nor is it universal within the legume families. I'll show you in a second. Question that's been asked for many years is, what is the nodule exactly? Well, we can look at it and see what it is. It's on the roots, usually the roots, sometimes the shoots of a plant, but it's, where does it come from? And so Ann Hirsch at UCLA and Tom LaRue, who used to be up the road here at Boyce Thompson Institute years back, asked, is it a modified root, a modified stem, or a completely novel organ, an organ sui generis? Early ideas about nodule origins included horizontal transfer of different components, such as leg hemoglobin. And those of you who eat Impossible burgers may be familiar with leg hemoglobin as the red color that you're seeing there, that substitute for myoglobin and hemoglobin. Anyway, so I could say more about it. I just gave this talk for Mark Sorrells in the background there, the polyploidy talk yesterday for your class, Mark. And one of the students pointed out that when you look at the Impossible burger, what is that stuff there in a real burger? Is it blood when you get the red stuff? And there was a big debate about whether it was actually blood or not. And it turns out someone looked it up and it's myoglobin. It's not hemoglobin. It's not blood that gives you the juice of a hamburger, apparently. But I didn't know that. On this burger, it's leg hemoglobin, so it's a vegetarian item. Anyway, to get back to the talk, the evolution of root endosymbiosis involves this nodule structure. And Martin Parniski and Katerina Markman asked this question, how novel really are nodules? And so this gets back to the original question about what it is. What is a nodule? So it turns out that most everything that is happening in nodulation is a matter of recruiting a pre-existing function or structure from some other program already present in the plant. And so my colleague Makoto Hayashi and Takashi Soyano asked this question and reviewed this some years back now. And so basically what nodulation has done is recruit gene regulatory modules from arbuscular mycorrhizal symbiosis, from lateral root development, from nitrate responses, and maybe from other places as well, from the whole infection process. There's all sorts of things, but almost nothing about nodulation is itself novel, the genes or anything else. So here at the heart of it all is something called NIN. And this recent review paper, NIN is a transcription factor called nodule inception. And so if you look at what happens during nodulation, you have the rhizobia being signaled by the plant and exchanging signals, infecting, starting an infection thread growth, proceeding into the cortex, initiating nodule formation, and eventually forming a complete nodule with this outgrowth from the plant, and eventually then becoming functional, involving in nitrate responses and all that. So every one of these steps is controlled by NIN. And so basically NIN, first of all, had to acquire some sort of binding sites for key elements of the signaling process we recruited from mycorrhizal symbioses, including the cycloidea binding site, which you see down here. And then the other thing that had to happen was that NIN targets must have acquired NIN binding sites. Now, there are about 145 direct interactors of NIN, at least one, that's one estimate. It may be an overestimate, but still a lot of different genes have had to come directly under the influence, the control of NIN. So all of those had to acquire binding sites from NIN. And so one of the questions, a big evolutionary question, is how, in the evolution of novelty, is how do these kinds of binding sites, if they're truly novel, how do they evolve? Even if they're not truly novel, how do they get brought into the control of a novel transcription factor system? So this is a major question. We'll return to that in a bit. So I haven't told you about polyploidy yet. The polyploidy angle here is that people have speculated for many years that polyploidy within the legumes, which had been known to exist, to occur, had been implicated in the evolution of nodulation. So this is the Medicago genome paper, 2011. And when the mitochondrial genome, or sorry, the Medicago genome was sequenced, it was found, not surprisingly because of previous work, that the genome was structured showing evidence of a whole genome duplication and shared with other legumes as well. So when soybean was sequenced, it showed it had that whole genome duplication as well. So looking at this, people speculated, well, okay, so you've got a whole genome duplication at the base of this whole group of legumes. We know that whole genome duplications can spur novelty. Nodulation also seemed to be unique to legumes, or at least probably evolved there as one of its sources of evolution. And so the speculation was, well, okay, so this is what's happened. We've had polyploidy either causing nodulation to occur or doing something to modify it if it already existed. So about the same time, this paper from René Gertz and his group, whole genome duplication has occurred at the root of the legume, Papilionoideae. This is the bean-like subfamilies of soybean and lupin and things like that. We hypothesize that gene pairs originating from this duplication event have evolved symbiotic functions. And then a couple of years later, another paper came out talking about it may have led to enhanced root nodule symbiosis in the Papilionoideae, the bean-like legumes. And you see evidence of that. This is a paper from some other colleagues of mine that came out just recently, looking at flavonoid synthesis and biosynthesis genes in the legume family and showing, again, that there's some novel genes found only in the legumes. Many of these are due to gene duplications and that are unique to the legume family. So this supports the idea that, that whole genome duplication has led to increasing the repertoire of genes available for nodulation. So I mentioned already that it's not necessarily found only in the legumes. So in fact, nodulation is phylogenetically scattered. And until 2018, which I'll explain in a moment, this was explained by a combination of convergence and loss. So it turns out that nodulation, once thought to be widely scattered in the flowering plants, early on in the era of phylogenetics was shown to be concentrated in one clade within the flowering plants. And this is called the nitrogen fixing clade or nodulation fixing clade. And within this clade, though, you'll see that it's scattered. So the light blue or dark blue here that you see, dark blue means that entire group has nodulation symbioses. Light blue means that it's scattered within that group. So in legumes, for example, the green is rhizobial symbiosis. These other ones are actinorhizal symbionts. It's a different group of bacteria. Within the legumes, you can see, again, dark green means that that entire group, every one of those plants will nodulate, with one or two exceptions where it's been lost. But in other groups, you'll see light green, and that means that it's scattered within those groups. So you have this distribution where everything is concentrated in one clade, but then within that entire group, you have origins or losses. It's just a very complex pattern. And again, nodulation is phylogenetically scattered. And mostly it was explained as a combination of convergent gains and then independent losses. The question was always how many of each. So we were pretty confident that there were multiple origins, but we didn't know how many and how many times it had been lost. So why do we care about this? I mean, I care because I'm an evolutionary biologist, and so this is certainly an interesting question, sort of curiosity-driven research question. But the bigger picture for people who are interested in applied sorts of things is that generating fertilizer for agriculture is a very expensive, ecologically expensive, as well as monetarily expensive proposition. And so wouldn't it be nice if we could engineer grasses, for example, crop plants that don't do this symbiotic nitrogen fixation, to have these kind of partnerships, make nodules, make their own fertilizer, essentially. And so if you're thinking about that, you might ask the question, how easy is that going to be? And if it's in evolution, it was happened multiple different times, that would be an easier proposition, perhaps, than if you it had only happened one time ever in all of the history of flowering plants. Of course, even if it's happened multiple times, it's happened only in that one clade. So something is unique about that clade that we have to figure out if we're going to make grasses do this kind of thing as well, right? And that predisposition, people called it, has been really elusive. Nobody has figured out what it is that makes that clade different from everything else. That's sort of a holy grail kind of thing as well. All right, so chasing unicorns, why do I say that? Well, because everybody kept talking about this, but it was very hard to figure out how you would actually test these different hypotheses. And so proving or testing whether or not nodules across different nodulating species are homologous. In other words, they have the same origin. They come from a common ancestor. That was the big question. How can you tell that apart from a convergent gain, right? So one of the things we thought about, this was following other work, it's not unique to me, my application to the legumes or to nodulation was maybe unique, but the idea was out there in other areas. Might the genomes of non-nodulating taxa harbor fossil evidence that they once nodulated? So that would help us to distinguish between primary absence of nodulation and secondary loss. And focusing on non-nodulating species instead of nodulators. So instead of asking whether nodules are homologous, we could ask for evidence that some plants that no longer show nodulation had actually once been able to nodulate. That's what we're trying to do. So in 2018, that was a seminal year for this, 2 different groups came up with tests of this, and both of them came to the same conclusion. And this first paper discovered signatures of multiple independent loss of function events in the indispensable symbiotic regulator nodule inception, NIN, and a number of different wholly sequenced genomes. And in this paper, taken together, results challenge the view that nodulation evolved in parallel, raises the possibility that nodulation originated approximately 100 million years ago in a common ancestor of all nodulating plant species, and then was subsequently lost in other groups. So this was great, and NIN was almost the only gene lost. There are a couple of other good candidates that sort of are lost here and there, but basically NIN is the one thing that's really unique about nodulation in terms of genes. So what did this look like? Well, here's a phylogeny. Here's the single origin of nodulation, the stars or losses. And here is, let's see, I think I've got an arrow for NIN showing here, this is the presence or absence of NIN. And if you look down this column and compare it with nodulating and non-nodulating species, you'll see that there's a very good correlation between the presence of NIN and nodulation, or the absence of NIN and non-nodulation. And so here's, again, what things look like in the genomes. You've got in legumes, vigna, phaseolus, soybean, pigeon pea, chickpea, things like that have all got a functional copy of NIN. Nisolia, shown here, is a legume that has clearly lost the ability to nodulate, and it is lacking NIN entirely. So a good correlation between a known loss and the absence of NIN. Here is a plant that's in the papilionoid family, but near the base, and it might or might not have lost it secondarily. We don't know, but the data here suggests that NIN is just gone completely, so it's probably a loss. And up here is the redbud tree, which those of you may not know, it's a legume. It's in the bean family, but it's one of the first offshoots that its subfamily is sister to the rest of the legume family. And in Cercis, the redbud, NIN is present, but it's a pseudogene, so it's been inactivated. So again, it's not always lost, but inactivation also shows you that this critical gene involved in nodulation is present in a non-nodulating species, but it is just non-functional. And we followed up, Shufeng Cheng is a good colleague of mine, friend, and he has been also looking into this further, more genomes sequenced. We see the same pattern whenever that happens. strong correlation between absence of NIN and lack of nodulation. So Robin van Velzen and Rene Gertz led this little essay type thing in Trends in Plant Science. Here's again the pattern that we're seeing. All these black things here, black clades, are places where where nodulation does not occur. And again, we're saying a single origin of nodulation followed by massive parallel losses. So the weakest link in any argument about having a single origin was always it took so many independent losses to account for this that it was never going to be a parsimonious argument. And as long as parsimony was your criterion for judging things, this just was never palatable at all as an ongoing argument. So how does this new hypothesis deal with that? And the answer is, so you're asking what could cause so many independent losses? And it has to do with atmospheric CO2 levels. And so what has happened is that global atmospheric CO2 levels have generally been decreasing in the last 100 million years since the time when nodulation is hypothesized to have risen. And consequently, because nodulation really requires a lot of carbon in the form of making the nodule, but also giving photosynthate to the bacteria in exchange for the nitrogen they produce, when you have a situation in which CO2 levels are decreasing, carbon becomes increasingly expensive, and the cost-benefit ratio between nodulation, nitrogen fixation, and the expense of the carbon changes and shifts. So you start out with a carbon to nitrogen cost-benefit ratio. It's positive at the beginning of this whole era, but it declines as CO2 levels decline in the atmosphere. It becomes more and more expensive. And so what the hypothesis says is that as that happens, different genotypes, different species, and remember this is a genotype of the plants by genotype of the bacteria by environment kind of proposition, G by G by E. As that happens, carbon becomes too expensive and nodulation ceases. So the plants just shut it off. So absence of nodulation is not loss, at least not initially. Right? So nodulation is plastic. And actually, if you look today, if you look at field legume crops, if you fertilize that field, the legumes don't bother to make nodules. It's too expensive. They get free nitrogen from the fertilizer, so they don't need to do that. So it's a plastic response. If you keep everything turned off after you've ceased to do nodulation, however, everything starts to decay as you remove the genes from selection. And so purifying selection is relaxed, and you'll start to accumulate mutations in these key genes. So that's why Cercis, for example, the redbud, still has NIN, but its NIN has decayed. It doesn't function anymore. Other plants have lost NIN completely. So this is what happens also with cave animals. Cave animals, when they go into the darkness, the eye becomes dispensable, and usually mutations occur pretty quickly to eliminate the eye. And there even are scenarios that are suggested somewhat controversially that involve positive selection, because even the machinery that's involved in making eyes, even if you're not using them, is also expensive. And so there may be some selection pressure to actually get rid of the eyes entirely. So this single origin massive parallel loss hypothesis became very popular in the community. Actually, in those two papers that I cited before, that's like 90% of the top players in the nodulation community were involved in one of those two papers. So people really are pretty invested in this hypothesis. But then, as I say here, this paper came around. Now, it's been in bioarchives for a couple of years. It took it a long time to get through, but finally published just a few weeks ago, actually. And so in this paper, what they did was they made a phylogeny of 12,000 different species of nodulating and non-nodulating plants in this whole clade primarily. Much of it from herbarium material, by the way. This is a shout out to the herbarium, because you can use herbarium material to actually isolate DNA and use that in a study like this. They updated the nodulation database of which species nodulate and which ones don't. They then used hidden Markov models to test the fit of different evolutionary transition rate models and reconstructed ancestral character states. So this is state-of-the-art phylogenomics, basically. This is what people do with big phylogenies. They test hypotheses like this. And they looked at root nodulation symbiosis. Whoops, that happened. Wow. And their results showed that the best models were 16 independent gains and 10 subsequent losses. So we've gone back to now the state-of-the-art hypothesis on a phylogeny is now saying 16 origins. The one that everybody in the nodulation community likes is one origin. So what's right? What's the truth? Okay. Now, truth and science, that's two words that you don't want to necessarily put together all the time. The question is, how do you test the homologies, right? We're back to that question, back to where we were before 2018, saying, okay, maybe there were multiple origins, but how do we test that now? And how can you test homology if recruitment was convergent? This is a big question. This is what's called deep homology. And I didn't mention that when I mentioned the recruitment process in nodulation, but those things that are shared across non-homologous origins, right, are themselves homologous. So lateral root development using scarecrow and short root, for example, that's something that's common to all plants, right? And that module is going to be homologous no matter how many different times it was recruited in different origins of nodulation. So looking at that is never going to help us answer that question, not just the presence of that, the use of that particular module, developmental module. So what if all the components are homologous? How do you test this? All right. So because what we really want to do is the uniqueness of the assembly that is what we want to test. So how can we figure out whether the same thing was built a couple of different times? That's the question we're getting at. So back to NIN again. Here's NIN, our friend. Are NIN binding sites homologous across putative origins? Remember that NIN had to be recruited. The targets of NIN had to recruit this binding site, all these different genes that you see here, right? Had to independently, in that model, in a 16 origins model, 16 different times, all of those genes had to recruit NIN function. All right, so how do we tell that? Well, can fossil NIN binding sites be detected in non-nodulators? So here's an idea. This is a paper we're writing now. It's sort of not a rebuttal to that new paper, but really just exploring how we can actually test these hypotheses. The people involved in writing that paper, by the way, are the best man at my wedding, and my academic grandson is the main author, and so it's kind of I'm not angry with these people or anything like that. I just want to get to the bottom of things. So NIN binding sites is positional criteria for homology. Imagine a gene. Here's a gene X in an ancestor, XA. It has a couple of different binding sites for different transcription factors. Now NIN comes along and this gene becomes recruited as part of the nodulation process and binds NIN. So here you have a NIN binding site. I've conveniently put it between these other two sites. This is in nodulating species 1. Suppose we sequence another nodulator from a different group of plants and we find the same position. That would be consistent with homology of that site. It's the same sequence, 8 to 15 base pair, more or less binding site, and it's in the same position. But if we sequence some other different genomes from other plants and we find that these NIN binding sites, even if they are the same site, because that's what NIN binds across different groups, if they're in different places, that would be consistent with non-homology. So that's the idea. There's not a lot to go on here, but this is about the best I think we're going to be able to do. And so we certainly know NIN binding sites. This paper just came out I guess a month ago or so. I just saw it a month or so ago. And anyway, there are NIN binding sites. Some of these interesting sites here are found across both nodulating and non-nodulating non-legumes as well. So they exist. We don't know the context. I don't know the context yet, but that's something we can look at. Okay, so philosophical questions continue to be raised about the nature of homology. So for those of you who are evolutionary biologists, have been hearing the word homology forever, since you first started reading about anything about evolutionary biology. And it's a major controversial topic, how to define homology and what it means and how to test it and everything else. Günter Wagner, who's an author on this paper, has been writing books about this for many different years. And here's character identity mechanisms are something that this group of authors has come up with, and I can't explain them to you because it's more philosophy than it is biology, actually, I think. But there's something that integrate information, come to this kind of hourglass structure. This is where the mechanism sits, and then has different readouts from that. The best example I can give you is actually for the origin of cell types. And this is another paper. This paper from 2016, Günter Wagner was the last author on this paper. The idea being that there's this integration mechanism here, the core regulatory complex, that takes all of the inputs, synthesizes that, and reads it out to coordinate the efforts of all the different transcription factors involved in this regulatory complex. And that combination, combinatorial factoring of transcription factors and their outputs is what makes a cell type specific to itself. It is the defining feature of a cell type. I guess I should say it that way is better. All right, so that's the classic example of one of these character integration mechanisms at the level of the cell. And then they talk about in this paper things at higher hierarchical levels, again, being analogous to that. So why do I mention that? That's my next thing. First of all, here's Giles Oldroyd's paper again. This is one you saw already, but they summarizes this very nicely. Rather than novel emergence of new genes, the evolution of nodulation appears to have involved the re-networking of pre-existing processes, principally controlled by the master regulator NIN. I told you all of that already. He goes on to say, they go on to say, we need to understand not only how this happens, but also how it's able to activate such diverse development in the different cell types. So now we're back to cell types. Not only do we need to know about cell types philosophically, but we need to know about them practically speaking to actually get to the bottom of this mystery. And that brings me to my last vignette, which is gene duplications and the origin of cell type diversity. So Jeremy and I have been interested in cell types for a little while, mainly because it turns out that the effect of polyploidy varies with cell type. And so some cell types respond by becoming much larger. Other cell types, on the other hand, don't. And so being as cell size is one of the key factors involved, we think, in pretty much everything downstream of polyploidization and in creating novelty. This is an important thing to know. And so we asked the question, well, first of all, what's a cell type? And guess what? People don't know, don't agree on what a cell type is. So this is 2022, 2023. Honghui Zhang is the head of the Allen Brain Institute, where they work on neurons and neuron cell types, where there are hundreds of, if not thousands of cell types. These authors are tops in their field. What's a cell type? This paper was just from a week and a half ago, and it's a little review paper, not a review paper, a little popular piece in Nature. And so I was actually interviewed for this paper. But so it's still a question of what this actually means. What is a cell type? And so, yeah, I was interviewed because I've written a little bit about it, because I was trying to figure out what in the world a cell type was and how to deal with it. Likened it to species, which is a very bad thing to do because species, nobody agrees on what a species is. And worked with Mark Lebo, so you'll see him in a second again, to just talk about this in a recent paper last year. Mark is one of the leaders in the field of single cell biology in plants. And I'm fortunate to be a colleague of his on a grant that we have together, along with Jeremy and Andrew Farmer, whose name you saw in relation to glycine. This is a talk that we gave this summer, actually presented at our botany meetings in Michigan, single cell perspective on duplicate gene expression. And this work is asking, this is an ongoing project, we haven't published anything yet on this, in soybean at least, to what extent does cell type and cell state diversity provide niches for duplicated genes? So what do I mean by that? So duplication of genes is really frequent. If you look at this, this is a classic paper here, Lynch and Connery 2000. You're looking here, the number of pairs, percentage of pairs of duplicated genes as a function of time. Right, so this is KS down here, so it's a proxy for time. This, you'll see this kind of L-shaped pattern in all different taxa. What this is, these are small-scale duplications, so single genes or a few genes at a time. And what this is showing is that duplication is frequent. You have lots of pairs at time t equals 0, essentially, and not much else beyond that, meaning that these things are duplicating, turning over copy number variation in gene copy number, and then loss almost immediately, okay? Now, Arabidopsis looks a little bit different, and that's because this peak down here is a whole genome duplication that Arabidopsis, the most recent of the whole genome duplications that Arabidopsis has experienced. So it's a little bit different, but the same idea occurs. You have a whole genome duplication followed by lots of losses of genes in that diploidization process I mentioned at the beginning. So we have to account for why gene pairs get kept ever, because most of the time, you think about it, these are redundant genes at the time of their formation. You don't need them. The plant was perfectly happy with just one copy before. And now we have to explain, why do some get kept and other ones don't? You've probably heard of neofunctionalization. This is partitioning expression by function. So a gene that had a single function, this duplicate copies have the same function initially, but then one of them acquires a novel function, and that gets maintained. Once these genes are partitioned by function, there's reason to keep both copies, essentially, being a little anthropomorphic here. So that's partitioning by function. Subfunctionalization is also involving function, but usually we think of it as being partitioning of expression involving location. So in this model, a gene has multiple functions, and then these are partitioned such that one gene is doing some of the functions, some doing the other. This diagram is not as good, I think, as this one here. This is a little simpler. And if you think of something that a single copy gene was expressed in limb and brain, and then after duplication, one of the copies loses the ability to be expressed in the brain, the other loses its ability to be expressed in the limb, now you have an indispensable two copies. You need one of each of those copies, and they're maintained. Okay, so this is involving location of the expression as much as anything. So looking at expression of location at the single cell level, we have bulk tissues, cell types, cell states of a single type, and then single cells within that. We have a gene duplication, A and B copies. We can ask about expression, whether one or both or neither are expressed. And if we look at the bulk tissue, the root might be expressing one copy predominantly, the leaf the other. If we now look at the root expression at the cell type level, we see the same kind of pattern where some cell types or cell clusters are expressing some, cell types are expressing one gene, some the other, some both, some neither. And now we break out the epidermis cells, for example, and look at these as they develop, becoming mature. Again, the same kind of pattern. And even at the single cell level, we may see variation in terms of which gene is being expressed. This level of the single cell is probably beyond, kind of for various reasons, beyond our ability to really work with very clearly. And so we're really talking about cell clusters, a mixture of state and type. A couple of years ago, we looked at expression and partitioning of duplicate genes in Arabidopsis, 11,000 different duplicate gene pairs. And this, we classified them, as you can see here. If neither gene was expressed in any of the cell clusters, it was a class 0, only one copy expressed, and that's what we see here in copy 1 in class 1. Both copies expressed, but always both were present, no fixation in any of the cell types. In this class 3, we have both copies expressed, and in some cell types, only one was expressed. In other cell types, both were expressed. And then here we have a case of what really looks like subfunctionalization. We have partitioning of expression in different cell types or cell clusters between the two gene copies. And this is what this looks like, one of the subfunctionalization cases. Your two genes that are derived from the whole genome duplication in Arabidopsis. And you can see here, one of them is expressed here, and this is endodermis. And down here, the other copy is not expressed there, but it's instead expressed in a couple of different places as well. So this partitioning has happened, presumably following gene duplication. We're now looking at soybean in this project at 10 different organs, and you can read the details over here, 120,000 single nucleus sequencing, 187 cell clusters, a lot of annotation. This is a project involving Mark Lebo and some of my other colleagues. Looking at 60,000 duplicated gene pairs by the type of duplication, and you can see the numbers here. The main thing I want to show is that this involves also two whole genome duplications. One that I've mentioned before is found in all the bean-like legumes. The other one is a much more recent duplication, only within the last, say, 10 million years or so, and that one is in soybean only. The other one is about 60 million years old. So if we look at the evolution of expression patterns following duplication, what do we expect to see? Here's a single copy gene expressed in a number of different cell clusters, or a number of different genes. We make a very simplifying assumption that everybody makes, unfortunately, and that is that immediately following duplication, duplicate copies are identical in their expression patterns and their functions. And there's some logic to that, but it's a pretty tricky one to make. But everybody has to do it for evolutionary purposes. There's not much else you can do because we don't have access usually to the progenitor species. So following duplication, you have two copies where you had one before, and if you follow that assumption, they are all doing the same thing, which means they're going to be class 2. That's what this is showing here. And that's at time t equals 0. If you let this go further, 10 million years or so later, you now have some of them still maintaining this class 2, but other ones have evolved to different classes depending on which genes are being expressed in which tissues. Some are silenced, some are retained, some might be lost entirely. So that's what we have for that prediction. And so the expectation is that over time, we should see a depletion of this class 2 type of gene where both are being expressed. Well, what do we actually see? Here's some data. First of all, class 0 is the one that's not expressed. These pairs are not expressed at all anywhere. So in every organ, most gene pairs have at least one copy expressed. Most of the genes, the largest single fraction are these class 2 genes, 20 to 30,000 pairs where both genes are expressed. We have only one duplicate copy expressed by loss or gain of 7 to 12,000 pairs, some fixation of one copy, and it's always the same copy in this case, 7 to 15,000 pairs, and then that little tiny bit up above here, expression partitioning within organs, and this is what that looks like, a few hundred genes at the most. And I'll just call your attention to the nodule because I'm going to loop back a little bit to nodulation here at the end of this talk. So basically, there's differentiation in the location of expression for about 50% of gene pairs after their duplications. So if we want to look at this time course, we can compare the old whole genome duplication, 60 million years, with the 10 million year duplication. And our expression is that there's a depletion of class 2 genes over time. And what do we see? This is exactly what we see here. If you look, there's much more of this class 2 gene in the recent duplication than in the old duplication. And we see this also in terms of the expression of subfunctionalization as well, this class 4. All right, my last thing here is to talk about co-expression networks. This is function now. And gene regulatory networks are involved in function. I think everybody would agree with that. And gene co-expression networks are proxies for gene regulatory networks. That's the point that's being made here. Chandler, who's a postdoc with Jeremy Coate, looked at this, reconstructed GCNs for all these genes. Here's some data, 400, 800 modules per organ, 40 to 100 genes on average per module. And the question here is, how often do both members of a pair belong to the same module, right? So what we're saying here is, and this is from the Arabidopsis interactome paper some years back, initially, following duplication, all the interactions of a gene that was single copy are maintained by both copies. So again, this is the idea kind of that I mentioned before, or location of function. But over time, we have interaction rewiring, and these two genes can now belong to different networks. Okay, so what we found was that 80 to 90% of duplicate genes in this data set belong to different modules. So a massive amount of rewiring has gone on across the board. So extensive functional differentiation of duplicate copies. And looking at this, thinking of this as a time course, we have the module in which one homolog, one of the two duplicated copies exists, and the module in which the second copy exists. Initially, we should see them belonging to the same modules, and all the genes would be shared, no change at all following duplication. And over time, we should see migration away from this nice diagonal into the other parts of this module space that we see here. What we actually see is pretty much that. Note the much stronger diagonal, still not great, but stronger certainly than this here, in the recent duplication versus the old duplication. So we're seeing that process of migration away from being in the same modules over time. This is what these modules look like, both duplicates in the same module. This is one of the nodule modules. It's hard to say that, but and here's nodule module 108, which is just the opposite, where only one duplicate is present in the module for most of the genes, right? And to come back to this, the nodule is a novel organ, as we said. And you might think that a novel organ would provide more niches for expression subfunctionalization. And in fact, that's more or less what we see. If you look at this, look at the comparison of the whole genome duplications, two whole genome duplications, the nodule is always on the high end, at the highest one in terms of the number of subfunctionalized cases of gene pairs that we see. Nodules do not appear to differ in the amount of partitioning that we get. It looks pretty much like everything else, but if you look at cell type specific modules in these GCNs, if you look at everything else, all the other organs look fairly similar here. This is the cell type specific modules here. The nodule is very different in terms of that. And why might that be? It is because the cell clusters that are showing the most of that sort of effect are actually novel cell types, infected cell types within the nodule. So, all right, so what I've shown you for this last part is cell types and states within organs provide apparently inexhaustible niches for refinement of duplicate gene expression. Single cell resolution allows us to track evolution of cell type specific expression over time. And the nodule, which is a relatively young organ, provides insights into the evolution of novelty and the role played by duplication in fueling innovation. So there are my three vignettes. Polyploidy generates diversity through speciation and genome evolution. It may have been involved in the origin or refinement of nodulation. And then these wondrous cycles of polyploidy can be observed in the occupancy of expression niches at the single cell level by duplicated genes. And with that, thank you.
Questions? So I had a question about essentially like synthetic biology with this NIN group. So has there been any attempts, to your knowledge, of taking those groups that because they would be closer, like the ones that do have the motifs for this NIN binding, but their NIN gene or set of genes is deactivated? Has there been any attempt to sort of reactivate that and see if they make nodules, or conversely, or I guess like in addition, been any more difficult aspect of like engineering this NIN plus motifs into another?
Yeah, so there's a lot of questions there, right? The first answer is no. Nobody has reactivated a non-nodulating species by putting a functional copy of NIN in. I'm guessing that Renee Gertz has probably tried that with Trima. This Parasponia group is actually in the cannabis family. It's a weird plant that grows in New Caledonia. And anyway, it has close relative Trima that doesn't nodulate. And so they're really closely related. So either it's a very recent gain of nodulation or a recent loss of nodulation, depending on which way you go. That would be a wonderful system for trying that experiment. I can't believe Renee hasn't done it, but it's not reported anyway. So let's see, other questions. Using NIN in other ways. People have actually shown that you can take the NIN from one nodulating species and go all the way to some other clade, completely put it in, and it'll compensate for mutations. In Lotus japonicus, for example, you can actually put NIN from this Parasponia, that cannabaceous thing, and put it into a mutant of Lotus japonicus and get it to function in nodulation again. So they are really interchangeable. As for looking at the binding sites themselves, people have done DAPSeq and things like that, trying to find where the sites are and what's interacting and everything. But the noise level in these things, Makoto Hayashi, I've talked to him about it, and he said they've done the experiment. They've got thousands of hits. And he thinks that even the 145 direct interactors with NIN is probably an overestimate. And he said other people are looking into this to really refine that more. So it's pretty complicated, but certainly people are interested in doing that kind of experiment. Randy, I think you had your hand up.
I never thought about the cost of running a nodule. Have people done an economic analysis and like yield per dollar and what it costs to
Yeah,
corn, for example, nitrogen fixing versus
right.
fertilizer?
That's a good question. I haven't seen an economic analysis of that. But people have, I mean, there's a lot of sort of information around that in a sense. Like, for example, that someone did a really neat experiment of trying to replicate carbon atmosphere from several million years ago and showed that nodulators would have had a much bigger advantage then than now, than in more recent carbon atmospheres. So that's economics, but of a different kind than you're talking about. Actually, what people are looking at now with engineering, Giles Oldroyd, who mentioned a couple times here, he and others have been thinking about what are the components of nodulation short of making a nodule. Could you get into grasses and actually have that be, a little more realistic to try to do that than get a whole nodule on the plant? And there's some neat things that people are finding. Jean-Michel Annet at Wisconsin found, I mean, he's not the one who found it, I guess, a land race of maize in the mountains of Mexico where that accumulates biofilms of nitrogen-fixing bacteria on the roots and actually derives a serious benefit from that. So that's another thing, another angle people are looking at. So no, Randy, I'm sorry, I don't have an answer for the dollars, but yeah, okay.
I'll just take the dollars.
Just take the dollars and run, Gaurav. And then.
Yeah, so regarding the last result about duplicate gene diversification, so we also see similar results in enzymes as the Ks increases, the correlation decreases. But I mean, it makes sense, but the alternative explanation is that duplicates that tend to diversify quickly are the ones that are selected and are retained for longer time. Duplicates that have overlapping functions tend to get lost.
Mm-hmm.
I'm not sure how to distinguish between that hypothesis and over time the divergent hypothesis.
Yeah, well, there's a lot going on under the hood, right, with those curves. As you know, because you know a lot about polyploidy, one of the other mechanisms that actually preserves genes in polyploids, but not in single duplicates, is dosage balance constraints, right? So there's a lot of nuance, basically, to those graphs that I don't get into here at all. In fact, that's one of the things we're exploring is can you see correlations between dosage-sensitive genes versus preservation? And there's a whole lot of things. We did that in the Arabidopsis paper to some degree, and we'll probably do that again here. But yeah, there's a lot happening with that. Laura.
I was just thinking a bit more about these assumptions underlying the gene duplication.
Yeah.
Have those assumptions been experimentally tested?
So Laura's asking whether the assumptions of shared or preserved function at duplication, could those assumptions be tested? I'm not sure you even need to know that they're really wrong in some cases, frankly. I mean, so for example, I didn't get into this in the seminar, but if you think about the difference between just duplicating a gene right in situ versus transposing it to some place in the dark matter of the genome where you're around a bunch of transposable elements, there's no way that gene is going to be the same function, and yet it's considered a duplicate, right? The other one is a retrotransposon kind of situation, or when you actually you chop out the introns and everything, and you're going to put that gene in there, call that a duplication, and is that really going to have the same function? So I don't know. One other complicating factor is that when we talk about autopolyploidy, so when we talk about polyploidy, often people just make the assumption it's just duplication of the genome, right? But an awful lot of polyploidy, probably all polyploidy, involves hybridization, either in the systematics and evolutionary biology sense of being between two species, or in the plant breeder sense. I'm looking at the plant breeders in the audience here, who think of a hybrid as being between two varieties of maize. Right? So almost everything probably when polyploids originate involves some kind of a cross. Different genotypes are involved. Now to say in an era when we know that there's allele-specific expression differences in pretty much every gene, right? To think that those are going to be identical? No, probably not. I don't even think you need to test that assumption. It's a bad assumption, but it's the best we can do for something that happened millions of years ago. Yeah.
Okay, thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.